Thanks everyone for joining us for our February seminar on physical genomics. My name is Benjamin Keene. I'm here at the Center for Physical Genomics and Engineering at Northwestern University. We are super excited to welcome our special guest this month, Professor Steve Henikoff. Dr. Henikoff is Professor of Basic Sciences at Fred Hutchinson Cancer Center and an affiliate professor of Genome Sciences at the University of Washington. He's also a Howard Hughes Medical Institute investigator, a member of the U.S. National Academy of Sciences, and a fellow of both the American Academy of Arts and Sciences and the American Association for the Advancement of Science. He received the Genetic Society of America Medal in 2015, and last year he received the 55th Louis S. Rosenstiel Award for distinguished work in basic medical research. His laboratory performs research on chromatin and nuclear dynamics, transcriptional regulation, centromeres and cancer epigenetics, and develops experimental and computational tools for studying these processes. Dr. Henikoff, thank you so much for taking the time to share your work with us today. One quick note for our audience. We do our question and answers at the end of Professor Henikoff's talk, so feel free to type questions into the chat box as you think of them, and we'll address them at the end. Or when we get to Q&A, just turn your mic on or raise your Zoom hand, and we'll do questions then. If you want to learn more about the research and training programs here at CPGE, you can visit us at physicalgenomics.northwestern.edu. We've got an archive of our seminars and talks on YouTube. Our handle is at PhysGenCenter, and you can also follow us on X or Blue Sky. So at this point, Professor Henikoff, I think I can turn things over to you. Thank you so much again.
Thanks, Ben. Is my screen okay? Is it showing up?
Yeah, you're good.
Okay. So I'd like to thank you for the invitation to tell you about histone overexpression in cancer. Now, I'd like to start out by drawing your attention to this stripped-down version of the nucleosome core particle, where you can see it's a tight left-handed spiral that the DNA simply follows around, and this protects the DNA from damage, for example, during active processes such as replication, transcription. So if you don't have enough histone, you're going to have DNA damage. But what if you have too much histone? Is that also a problem? Well, if you're a yeast cell, it's not a problem at all. It actually extends your lifespan. But what if the same thing were to happen in a cancer cell? Could that be a hallmark of cancer, resisting cell death, enabling replicative immortality? And this is what we're going to talk about today. And we're going to talk about it in the context of formalin-fixed paraffin-embedded sections. These are standard pathology sections. So standard practice is for a surgeon to remove a little bit of tissue. It gets dropped into formalin at a concentration of 4% formaldehyde. It'll sit there for a couple of days to a week. And then it'll get dehydrated, embedded in paraffin, placed on a microtome, slices are cut up, picked up on a slide. They're stained. A pathologist will look at it under the microscope and say, give it a WHO grade, something like that. And then the rest of that paraffin block is placed into storage. And this has been going on for well over a century. So these blocks have been building up in hospitals and clinics around the world by the billions. And they present really great opportunities given all the metadata, all that you can use for retrospective studies, for example. And this has generated sort of a flurry of methods that people have applied, whole genome sequencing, RNA sequencing, for actually getting molecular information out of this. Problem is that formalin damages DNA and RNA degrades in storage. Now, I'd like you to think back to when you were maybe in high school or undergraduate and you learned about the central dogma of molecular biology, where information flows from DNA to RNA to protein. Of course, that's the product. I want you to think now about the machinery that does it. In the case of RNA, it's RNA polymerase II that makes all of our mRNAs and most other types of RNA. And RNA-pol II is a large multi-protein machine with a hole on one side where the DNA goes in, a hole on the other side where it comes out, another hole where the RNA comes out. Now, the problem comes from the histones. The histones are loaded with lysines, and so formalin treatment will heavily cross-link the lysines, and that'll basically turn the chromatin landscape into pretty much impermeable stuff. However, Pol2 is different, you see, because the largest subunit, the catalytic subunit of RNA Pol2, has a C-terminal domain that comprises about 52 tandem copies of a heptapeptide repeat without a single lysine, so it's immune from this. And this gives us high signal to noise difference between the phosphate on serine 5 that we can pick up versus the rest of the genome that's pretty much impermeable, okay? Now, serine 5 phosphate is a mark of paused RNA polymerase 2. It pauses before undergoing productive elongation, so that's a checkpoint, a critical checkpoint for gene expression. And that's what we're going to be looking at today. And the way we do this is we bind an antibody to the phosphate. We then bind a fusion protein between protein A, which binds to the antibody, and TN5, which is the cut and paste transposase. That's the basis for example, the ATAC-seq method. And then we activate the TN5 by adding magnesium. We then amplify the DNA and sequence it. This is what we call cut and tag. But we've modified it in such a way that we get high accessibility around paused Pol2 throughout the genome, and this will map enhancers and promoters throughout the genome, all the regulatory elements where RNA Pol2 is. Very similar to what you get when you do ATAC-seq, but we're looking at the transcriptional apparatus itself. And we do it in a way by doing the tagmentation at low salt. This means that the TN5 will actually bind to the DNA before we add the magnesium. So we're looking at Pol2. So you might think of it as open chromatin, whatever, same thing, but in this case, we're looking at the transcriptional apparatus, and that's going to become important as I go on. And we have a very simple way of doing it. We do everything on the slide. So we'll take one of these FFPEs. We will then put it in some buffer for cross-link reversal, heat it up, and then drain it, then add a little bit of antibody solution. We cover with plastic film, real simple stuff. And then we either dip to wash or squirt a buffer on it to do the wash. And we do that for each of the incubations, each of the steps, up to the point of tagmentation by adding the magnesium, after which we then scrape it into a tube, we amplify libraries and sequence. And this whole thing is really easy to do and fast, and it's cheap compared to the competing methods, RNA sequencing, whole genome sequencing, panel sequencing. And we only need about a tenth of the material. Now, when you consider that cancer screening, each one, it's in the range of thousands of dollars. This is very attractive to do this, to get all this other information that you otherwise don't get from when the pathologist gives a WHO grade. Furthermore, it's really easy to do, and we taught it this summer to 2 high school students, 2 undergraduate students. They did great. They learned about the central dogma, for example, and we actually recently published it as a guide for educators. Now, since we're doing this on slide, we might consider doing this spatially. And you might be aware that spatial transcriptomics is really quite expensive. Can we do this more cheaply? And this is work of Kami Ahmed, my colleague at the Fred Hutch. And what we do is we print out an XY array using a dot matrix printer, so oligos, splint oligos that we can use for ligation to the ends of the fragments after tagmentation. So after tagmentation, we just sandwich the 2 slides together with DNA ligase in between, and basically the splint oligos will get added on, and then we scrape it off and do the usual thing. And since we know the locations of the oligos, we basically do the usual UMAP kind of clustering, and then we can reconstruct without an optics-free reconstruct, we can reconstruct the tissue on there. Now, what we're looking at here is a mouse glioma. It's a mouse brain section, and it's a glioma, and we can actually pick out the glioma. Now, this is low resolution, you'll say. However, we're now switching over to hexagonal bead arrays with hundreds-fold more resolution, a good enough resolution that with this is work of Sanjay Srivatsan, my colleague at the Fred Hutch, and he's been able to basically use computational tools in order to get essentially single cell resolution. And we do this cheaply because these slides can be printed out, can be coded very, very cheaply. So we're looking forward to actually applying everything I'm going to tell you about spatially at high resolution, maybe single cell resolution. Okay, so that's the methodology that we're going to be using. And what I'm going to go through over the course of this talk are the various kinds of genomic features that we can get from it, beginning with regulatory mutation, promoter enhancer activity, microRNA transcription, hypertranscription, but focusing mostly on histone gene transcription. First of all, regulatory elements. Now, regulatory elements are discovered in FFPEs by whole genome sequencing, but work of my colleagues at Fred Hutch, Xu Lin and Zheng Guai, has been able to apply come up with an algorithmic approach to identify this in our CUTAC data. And the problem with whole genome sequencing is really twofold. First of all, because FFPEs, because there's a lot of mutations, you get sequencing errors, artifacts. If you only have one observation, then that's not really good enough to distinguish it from a somatic mutation from the artifacts and errors. So you really need to sequence very, very deeply. That's one problem. The other problem is that when you do whole genome sequencing, enhancers and promoters are GC rich, and because they're GC rich, they get discriminated against during the PCR. And so therefore, even though it's supposedly whole genome, enhancers and promoters are underrepresented in general. So what we can do here is, because we only have sparse coverage, we only get sparse coverage for what we're doing, we're more likely to have multiple observations so we can be confident. And now this just shows what our colleagues have been able to pick up in standard sections that we prepared. In this case, we have 7. So what I think of this as a random sample of cancers throughout our species. And I'm going to refer back to these 7 examples later. But first of all, I want to get, so that's the genetic side. So let's get to the epigenetic side, that is PAL2. So in this case, it was a matter of looking at a mouse brain tumor from a single mouse driven by REL-A transgene. And it was a matter of scraping 2 tumor sections and 4 normal sections into separate PCR tubes, and then basically just doing the standard protocol here. Now we're going to focus on candidates as regulatory elements. Now, these are from the ENCODE project and from putting together mostly DNase hypersensitivity mapping. So it's open chromatin mapping, and basically synonymous with what we're looking at here. So in our analysis, we don't look at genes. We're going to look at regulatory elements, which have enhancers and promoters, and are basically, instead of 20,000 genes or so, you basically get order of magnitude plus more. So it's information rich. And you can see that here, we can well separate the tumor from the normal. Now, if we look at a particular example here, the top 3 based on fold change are a collagen gene, and you can see the kind of signal and noise that we're getting is pretty good. And if we look at the other side, we see the inverse pattern here. Now, the top 10 correspond to something called MER124A on 2 genes, encoded by 2 genes on 2 different chromosomes. And MER124A refers to a 22 nucleotide microRNA. And so by far the most downregulated loci is this particular microRNA gene. It's actually known to be a tumor suppressor gene, well studied. Now, what we realized was that because microRNAs are very hard to pick up, so you don't pick them up in RNA sequencing data, particularly if it's they're very small and they don't have poly A on them, and so it's a real problem trying to pick them up. We realized that maybe this is a particularly good way for looking at microRNA genes by looking at PAL2 over the gene. And so we did that simply by looking at RefSeq annotated microRNAs and just aligned them to these 7 assorted tumor normal cancers that I showed you. And we see that these 3 come way up at the top of the list based on rank, and they come up pretty much the same in tumor and normal. And we wanted to confirm that, so we looked at some leukemias, AMLs, about 85 of them we looked at, and it turned out that we got the same 3 in the same order. So these guys really stand out. But what really caught our attention was that these 2 are in chromosome 21p. Now, 21p is the short arm of one of our 5 acrocentric chromosomes, 13, 14, 15, 21, 22. And there's not really nothing on there except for the ribosomal genes. All of our ribosomal genes are encoded on the short arm of these 5 acrocentric chromosomes. So it made us wonder, well, maybe these microRNAs are basically from the ribosomal genes. And we looked through the literature and only found one paper that really picked up on this. And it goes in a journal I never heard of. Basically, it goes back about 10 years ago. And they picked up 2 out of the 3 that we had found. The third one hadn't been annotated yet. And these 2, they identify as being in the 5' external transcribed spacer of the ribosomal genes. And they also picked up these 2, MER1275 and 6724, that are in the non-transcribed spacer. So what's going on here? So we basically just took a single copy of the rDNA and aligned and we basically confirmed what they showed back 10 years ago, where we see that MER1275, 6724 is in the upstream region, upstream of the 47S precursor, and the others are in the 5' external transcribed spacer. Now, this hadn't been seen because the genome assemblies that we and other people have been using for all along basically black this out because these are repeats, and nobody wants to deal with them. But if we look at the telomere to telomere assembly, which came out about 4 or 5 years ago, we see that in the CHM13 cell line that was used for the assembly, we see that these microRNA genes are 100% conserved in all 224 copies. Well, okay, are they actually expressed? Now, to look at expression, we looked at ProSeq data. So this is all published data. We looked at ProSeq data, where ProSeq maps the RNA base and the active site of RNA polymerase. Is quite good for looking at nascent transcription. Now, ProSeq doesn't care what the polymerase is. It'll get from all the different polymerases. So as you might expect, you get a huge ProSeq signal over the ribosomal DNA here. So that's here on chromosome 21. And this is 80% of the transcription in the genome, but everybody ignores it. Okay, so this is all PAL2, the little bit of stuff here on chromosome 21. Now, if we look at the telomere to telomere, if we look at a single, use the telomere to telomere assembly and blow it up here, so you can see that there are multiple copies in tandem. And you can see that in the 5' external transcribed spacer, the nascent transcript is basically gets chewed up, and then the 18S, 5.8S, and 28S RNAs get processed out on the nascent transcript. And then what we see is right upstream is this MER 1275, 6724. So let's take a closer look at that. It's the biggest peak. And it's perfect. That is, the 5' end of a 50 nucleotide transcript corresponds to the 5' end of MER 1275. The 3' end of 6724 corresponds to the 3' end of this 50 nucleotide transcript. Whether we look at ProSeq, which gets a 3' end, or ProCap that gets the 5' end, it is like basically perfect. And it's conserved in primates. You see exactly the same 50 nucleotide transcript in chimp and macaque. We also see conservation of the sequence. So MER12, the annotated sequence, MER 1275, is identical between human and chimp. And what we want to know, are these really microRNAs? And for that, we want to see what the precursor looks like. Does it fold back on itself as microRNA precursors must do in order to get processed? Now, microRNAs, their job is to base pair with mRNAs and target them for degradation. This is a level of gene regulation in between transcription and translation takes place in the cytoplasm. So we asked, do they show the foldback? Indeed, they do. This is using the RNA fold server. We basically see in this Vienna format that it folds back on itself with high free energy. So basically, these are ordinary microRNAs as far as we can tell. Okay, they're just made at a very funny place at a very high level. Now, there's more to the story about how they actually how they're regulated. They are insulated by CTCF. So there's an interesting story there. I'm not going to go into to stick with the theme today, but I'll put up a QR code where you can look at it. It should be published soon, but anyway, it's up on bioRxiv for you to look at for the rest of the story. So what do they do? Well, it turns out that 6724 has been very well studied. It's an abundant cancer biomarker. It's the most significantly upregulated microRNA based on hybridization microarray assays, the most significantly upregulated of the more than 2,500 in hepatocellular carcinoma. And another study showed that in bladder cancer, same thing. It's the most significantly upregulated microRNA. What about the other one? Well, it's also an abundant cancer biomarker. For example, it consistently upregulated in plasma, urine, and tissue. It's the top hit. It's probably the best cancer biomarker anyone has come up with so far. And you can basically pick it up in urine with very high sensitivity and specificity. So I was curious, and I just typed it into Google Scholar, MER 1275, and there are more than 2,800 papers that refer to it, mostly in cancer. And if we look at the common cancers, we see that it's found in all of these different cancers just based on citations. It's also found in papers on exosomes. Now, exosomes are extracellular vesicles, and what they probably are doing with respect to microRNAs is that the microRNAs, by regulating mRNA stability, basically are probably are thought to condition the local environment. So, so what I'd like to do is move on, and if you want to take a look at we have an interesting story about CTCF insulating this particular region of the genome. Anyway, you can look it up, use the QR code. But what I'd like to do is move on and ask, so why is it so high in cancer? And a very straightforward way of explaining it is something called hypertranscription, where all of the RNA polymerases go up. There's increased protein synthesis, increased ribosome biogenesis, and this is common in stem cells. It's a way that stem cells presumably become special by hypertranscription. transcribing, but hypertranscription has also been seen. So basically the idea is that these are already the highest expressed of the microRNAs just because there's so many copies of them. And when there's hypertranscription, they're going to go up to the top and become the best cancer biomarkers. Not that they're doing anything different from other microRNAs, just because they're hypertranscribed. Now, hypertranscription has been seen to be widespread in cancer. So, for example, in this study, they showed that it confers a worse prognosis independent of all these other factors. And this is based on pan-cancer data. Now, this was all done using RNA sequencing. And in that case, you have to know how many cells you're starting with, how much DNA there is to get the ratio of RNA to DNA. We don't have that problem when we're looking at RNA polymerase II because PAL2 is on the DNA. And so let's say that in a brief window of time, you have one PAL2 at a site. Then if there's twofold hypertranscription, there'd be two. If there are two, there'd be four. If there are four, there are eight. You get the idea, proportional upregulation of gene expression. And we can plot this, we can measure it in a very simple way. We simply subtract normal from tumor. They're just, doesn't really matter how we normalize them, just so they're normalized to the whole genome. And then we take the average, t+n/2, and plot it on the x-axis. So if there is a candidate cis-regulatory element, we have a million of them in a single copy in the human genome. If there's hypertranscription, if it contributes to that, it would be a positive number. Hypotranscription be a negative number, plotted on a log scale for clarity. And if we go through those 7 tumor normal pairs I told you about, we see that there's strong hypertranscription in the case of breast. And in fact, 6 out of the 7 of this sample from humans, basically of cancers, we see hypertranscription. Now, because we have such dense coverage of CCREs and PAL2 over the genome, we can identify focal amplification. So this shows up a region of chromosome 17 that's well known to be amplification of the HER2 cancer driver, well studied, particularly in breast cancer. And we see these 2 patients have HER2 amplifications. Now, it's easy to understand how HER2, which is a known cancer driver, if it's increased relative to everything else in the genome, that would drive cancer. But it doesn't explain how hypertranscription can help a cancer cell. Why would it do? If everybody goes up, who's the gain? Unless hypertranscription helps a cell during DNA replication. So think back to what I introduced you to at the beginning, where histones need to be made. They need to be made just in time. They are made just in time for packaging right behind the replication fork. And that protects histones, that protects the DNA as it's replicating. And we calculate about 4 to 5% of the total protein is going to be histone. They're small proteins. That's a lot of histones. And we can see that the histone loci actually are quite greedy with respect to getting RNA-PAL2. This is shown, for example, in this study from Christine Russell's group, where they basically looked at Drosophila embryos at the zygotic genome activation. And you can see that PAL2 is in these nuclear bodies called histone locus bodies, I'll say more about. And in a ZELDA minus mutation, where ZELDA is the master regulator of zygotic genome activation, it's as if all of the PAL2 gets sucked into the histone locus bodies. Here's another example from mouse embryonic stem cells from Phil Sharp's group. They did GROW-seq, think it's the same as PRO-seq basically, without the high resolution. And it looks at engaged RNA polymerases. And what they noticed was, since it's single cell, they could identify the cells that were in S phase because they have so much RNA-PAL2 over the histone genes relative to everything else. When they quantified this, they found there was a global reduction of 40% at the G1 to S transition, a global reduction in RNA polymerase overall, except for the histone genes, which is basically sucking it up. So what this suggested to us is that histone gene transcription is rate limiting for DNA doubling. This might give a cell, a cancer cell, an advantage at S phase, at the G1 to S transition, to go through the cell cycle faster, so it would have an advantage over its neighbors. How do we test that? And what we're going to do is take advantage of work of my colleague at the Fred Hutch, Eric Holland, where Eric has been putting together different tumor types, RNA sequencing data from multiple studies, and using them to predict outcome. And here's what he does. Okay, meningiomas are intracranial tumors. They're mostly benign, but they can recur and be fatal. So it'd be very important to be able to predict from looking at the FFPE of the meningiomas what is the likely outcome. And here's how he does it. So he takes a UMAP approach. So each of these dots is a single patient, and you see there's some bad places in two-dimensional space for WHO grade. But he can do a lot better by taking RNA sequencing data from every new patient coming in, and in multidimensional space, identifying the 20 nearest neighbors and using the known outcome from that in order to come up with a prediction as to whether it's going to be benign or malignant. So we thought that, well, maybe we could try this. So Dorju, who's the technician in the lab, went ahead and we got 30 meningiomas slides. They were coded, so we didn't know the outcome. And then when she was a postdoc in the lab, Yi Zhang went ahead and just looking at the amount of RNA-PAL2 at the histone genes, predicted that 5 of them would be malignant. And she couldn't have done better. Basically, those 5 turned out over a 25-year period based on nearest neighbors are predicted to be malignant, other 25 benign. Okay, but she also found something else, and that was that the histone, high PAL2 at the histone genes predicted aneuploidy. Now, aneuploidy is the first hallmark of cancer discovered by this guy back in 1890 when he was looking at mitotic figures and noticed that in cancer, but not in normal, he saw some chromatin hanging off the metaphase plate. Now, fast forward about a century plus, and the Cancer Genome Atlas tells us that about 80% of aneuploidies are whole arm gains or losses. Now, how do you get a whole arm gain or loss? Well, there has to be a break at the centromere or in the paracentric regions. And so basically what we realized was that we're seeing that whole arm gains and losses correlate with the total amount of RNA-PAL2 at the histone genes. And this is one example here. So we're looking at the 30 meningiomas here, and we're looking at chromosome arms going from chromosome 1p to 22q. These are the autosomal ones. And 38 out of 39 show PAL2 histone genes correlating with whole arm losses relative to gains. So the same thing in a cohort of breast cancers. And in this case, it's more significant, we infer, because these are all malignant, whereas only a fraction of these are malignant. And furthermore, we started looking through the literature and found that there's one paper that actually was onto this, although nobody cited it. But it turns out using gene set enrichment analysis after filtering the data to remove correlations, filtering the data using a linear mixed model, they found that histone genes were really very highly correlated, associated with aneuploidy in pan-cancer data relative to these other genes. Okay, so it's real. Now, we wanted to take this one step further and ask whether we, just looking at whole arm losses versus gains, whether we can actually distinguish, predict outcome in cancer. And indeed you can. In the meningioma data, the RNA-seq data, we found that more than 6 whole arm losses predicts unfavorable outcome, and fewer than 3 predicts favorable outcome. And then looking at pan-cancer data, just putting it all together from the Cancer Genome Atlas, we still see high levels of statistical significance, despite the fact that there's so much heterogeneity for, again, whole arm losses will predict, if there are enough of them, will predict unfavorable outcome overall. it's as if elevated pol-2 histone genes drives faster proliferation, so a cell has an advantage over its neighbors, and chromosome arm losses that can further drive cancer. And here's a very simple model that comes from work of others. First of all, centromere protein A is the epigenetic mark of the centromere. It's absolutely required for centromeric nucleosomes. It's an H3 variant, and it's absolutely essential for making a centromere. It basically, if there's a high concentration of CENP-A on a chromosome, it'll make a centromere regardless of the DNA sequence. And over 20 years ago, Gary Carpenter and colleagues had shown that if they overproduce histone H3, it displaces CENP-A in both Drosophila and humans. And others have since shown that if you overproduce, or if you knock down CENP-A in normal human cells, what'll happen is that you'll get centromere breaks, and the breaks are apparently caused by replication transcription conflict. So they occur at S phase during replication, and that's what breaks the chromosomes. So here's our model for what's going on here. If there's a centromere break and a chromosome arm loss, it can be lost or it can be gained depending on what happens when in the next cell cycle. But it can also get trapped in a micronucleus. Now, because a micronucleus has much less surface area than the regular nucleus, it can lose, this is what people think, it can lose protective factors that will then lead to DNA breaks and DNA damage. So here's our model, basically, that histone overexpression followed by aneuploidy. Eventually, you get a tumor that's big enough to see. This would begin in a single cell, and then the cancer will basically make these extra chromosomal DNA circles that people have documented, as well as the microRNAs to help drive the cancer. Now, it's been said that if you live long enough, you'll get cancer. So this raises the question as to whether histone overexpression, which really we think gets the whole thing started, is a feature of normal aging. And to look at that, Sarah Leichter, a postdoc in the lab, has been studying the Drosophila gut epithelium. Now, there are four different tissue types in the gut. What's important about this is regenerative tissue, the gut epithelium, because of the rapid turnover of cells over a matter of, in Drosophila, 4 days, in human about 7 days, basically. It means that we can look at flies of different ages, and the age of the cell is the same in all of them, even as the fly ages. I'm not going to go through all of this. It's been recently published, but basically what we see here by using a single cell approach for using a single cell approach with cut and tag of the histone modification H3K27 trimethylation, which is responsible for polycomb silencing. What Sarah sees is that there's a decrease in the differentiated cells, the enterocytes, and an increase in the stem cells. So we wondered, could it be that the stem cells are being driven by overexpression of histones? And so we did the same kind of analysis that I showed you before, but rather than subtracting normal from tumor, we're going to subtract young from old and do the same kind of thing. And what we see is there's overall hypotranscription, but the histone genes go way up, drastic increase as the stem cells age. Okay, so now that I've shown you what we think about how histones are driving cancer as a normal process of aging, it brings up the question of what regulates histone gene transcription. After all, that seems to be the key question. And I'd like to refer back to what I showed you before about the histone locus bodies and about how they're really greedy for pol-2. Now, we already know from much older work that the cyclin E CDK2 kinase, which is the master regulator of the G1 to S transition, phosphorylates NPAT, where NPAT is the transcription factor and the major component of the histone locus body. And it's sort of this is a artist rendition of the histone locus body, and you have this NPAT in green basically forms actually is a structural component of it, but it also interacts with the DNA. And there's a whole lot of PAL2 in there, but there's also the machinery for 3' end formation. I have to point out to you that the reason that histone genes tend not to be looked at is they're not polyadenylated. They have their own process, their own processing, which we can actually phylogenetically date back to the origin of eukaryotes. So they've been doing something, the same thing, for billions of years, but not using the standard spliceosome apparatus or anything like that. So that's part of the reason that they're not picked up. You have to study histones all by themselves. And the way we do this, and this is work of Kami Ahmed, is to take advantage of a very nice system that's been set up for studying histones, and that is to look in Drosophila. We can look into Drosophila testes and then do knockdowns of essential genes because the testes aren't essential for life, just for sperm. And they have a stem cell niche where there's a proliferative zone followed by exit from cell cycle proliferation into G2, into so-called gonial cells. They'll undergo meiosis and followed by sperm formation, but we don't care about that. We just want to know what's happening in the proliferative zone, going from proliferative zone to the G2 so-called gonial cells. So you can see that here. We're looking at phosphorylation of NPAT caused by cyclin E CDK2 to pick up the histone locus body. So you can see these tight little dots here in the proliferative zone. So the question is, what happens if you knock down suppressors, basically silencing the silencing machinery? So by looking at the usual suspects here, we basically don't see anything in knocking it down. So then Kami went ahead and asked, well, what about the histones themselves, including the variants? And what we see is there's one histone that has an effect, and it's histone H4. And here's an example here where we're looking at H3 GFP, normally the partner for H4, but basically if you knock down H4, H3 goes up. We see the same thing with H2A. So it implies that free histone H4 represses the histone locus body transcription. And we can see H4 directly over the histone locus body, called MXC in Drosophila here. And basically you can see that H4 marks it. Same thing in human cells. So here's human K562 cells, and you see that basically here's chromosome 6, the large cluster, chromosome 1, the small cluster, and basically we see that H4 is over all of them. And when we do cut and tag on NPAT and H4, now NPAT is the transcription factor that actually regulates, the sole transcription factor that regulates these genes, and it's bound right in between. And we see that H4 by cut and tag is directly over it. And we don't see it anywhere else in the genome, just in the histone locus body at the replication coupled histone genes. Furthermore, of all the human proteins that are predicted by AlphaFold, H4 is the top predicted interactor with NPAT. So what this implies is we're seeing direct negative feedback regulation by free H4. Now, Weifeng Wu, postdoc in Lab 1, to take this one step further. And she has come up with a method, a general method here for mapping the transcription bubble, that is this thing, in PAL2 specifically, by using pithoxyl, which will derivatize, covalently bind to guanines, but only on single-stranded DNA. And it's got an azide on it, so we can do click chemistry on it. And this gives us pull down and input after streptavidin and pull down. And if she looks at genes and asks where they fall on this rate of this ratio of pull down to input, basically the histone genes have the highest bubble density of all types of genes. So here's an example. If we look at a bidirectionally oriented H2B, H2A gene, and we look at NPAT and PAL2, we can't see any difference. They're basically one right on top of the other. Okay? And it doesn't matter whether it's the bubble or the input. Okay, and now when we look at all the histones, basically we see that they're just one right on top of the other, NPAT and NPAT and PAL2. And it doesn't matter what time, where we are in the cell cycle, we see the same thing. So this is throughout at the histone locus body. You have NPAT and PAL2, and they're just superimposed on top of one another. So here's what we think is going on. Now, NPAT is a highly unstructured protein, and you can see from the AlphaFold prediction. And what we think is going on is that the unstructured regions, as others have shown, unstructured regions of the CTD of PAL2 and the unstructured region of the NPAT will basically have weak interactions that will hold NPAT on there as PAL2 is just waiting. And then our model is very simple. We're basically saying it's the CDK2 kinase, which will phosphorylate NPAT. We know that, but also others have shown in vitro that CDK2 kinase actually is a better kinase for the CTD of RNA polymerase II than are the transcriptional kinases. So basically, it suggests that the repulsion basically of all the negative charges that you get will basically propel the PAL2 out there and release NPAT, and this will just keep going so fast that we basically see everything happening all at once in the histone locus body. Okay, finally, I'd like to close with the question of, okay, if H4 is silencing outside of S phase, what derepresses H4 at the G1 to S transition? And this turns out to be PRMT5. And this was shown in a study in bioRxiv from David Schechter, and PRMT5 will modify arginines by placing two methyls, it's symmetrical dimethylation of arginine, and it will target histone 4, arginine 3. And what they showed in the study is that using a PRMT5 inhibitor, they saw gene expression of the histone genes, just the histone genes, drop down within 15 minutes. They saw reduction in expression. They did the proteomics and showed that histone amounts drop within 15 minutes. Now, PRMT5 is well studied. Pharma's been all over it for well over a decade. There are multiple drugs that are in clinical trials because it's thought to be a central regulator, tumor cell biology, most promising anti-cancer target in the field. And I consider this strong evidence, since we didn't know anything about this when we came up with the histone overexpression model, as strong evidence in favor of our model that histone overexpression is driving cancer. Okay, to summarize, I've shown you that we can use our FFPE CUTAC method for PAL2 serine 5-phosphate to identify active regulatory elements in FFPEs, map microRNA cancer biomarkers, reveals hypertranscription in cancer, detects focal amplifications. I showed you that PAL2 and histone genes can be used to predict outcome in cancer, correlates with whole arm losses, increases as stem cells age, and the HLB is repressed by H4, histone H4, and derepressed by H3, arginine 3 dimethyl. And I'd like to make a shout out to members of my lab and also Eric Howland, our collaborator, who've participated in this project, particularly Kami Ahmed, my colleague at Fred Hutch, Yi Zhang, who is continuing this work in her own lab at MD Anderson, as well as Doris and Georgia and Weifeng Wu's work I talked about at the end. And I'll stop there and take questions. Thank you very much for your attention.
Thank you so much. That was beautiful. Looks like we've already got a question in the chat. If you can see that, I don't know if you just want to.
Yeah.
If you prefer, I can read the question.
Oh, I see it here. Okay. So the question is, do we see overexpression of specific histones that have effect? And we've looked at that, and I must say that it's been sort of disappointing. We were hoping that maybe there'd be less H4 or something like that, but we don't really see that. It seems as if the histone genes all of them, there's no difference that we see. I did point out, though, something interesting in normal aging in Drosophila that might bear on this, and that is that H2A, H2B were really high relative to H3, H4. We don't understand that, but anyway, maybe there's some clue there. But we haven't seen it in cancer, but we do see differences in aging. There's another question. Uh, Okay, so there's a question that basically comes down to what is actually normally regulating the histone genes. Now, to think back, we think of this process at DNA replication of filling in the histones as going back to the origin of eukaryotes, and so every cell, if you have 20, 30 trillion cells in your body, every cell has gone through this multiple times. So the regulation has to be really good at S phase in order to get the right amount of histones there. Because if you have too little, you get DNA damage. If you have too much, then you get something like what undergoes in stem cells in cancer. And so it's all under control. And we know from work of many groups that the level of histone mRNA is tightly controlled. So after S phase is done, then the excess histone transcripts get degraded. The other thing I didn't mention is that there are histone variants. So when a cell exits the cell cycle, when you lose a histone, you get the variants put in outside of replication. And for example, in brains, you no longer have any H3. It's all been replaced by H3.3. And these variants need partners. CENP-A needs partners. So the partner histones get made, and those are ordinary polyadenylated messages. They don't use the same process. So there's a whole level, there's been multiple levels of gene regulation at every stage that controls it. And it's really kind of understudied. So I don't really have a good answer for any simple answer for that. It requires more work.
I think we've got one from Dr. Foltz. Do you want to turn your mic on?
Yep. Hey, Steve. A beautiful, beautiful story, of course. So I have two questions. First, on the first section, when you see the upregulation or the increase in histone gene expression, are you seeing more polymerase at the same histone genes, or are you seeing more histone genes activated? Because as you know, right, there's a number of H4 genes, there's a number of H3 genes, even that all encode the same protein. I'm wondering if different genes also get activated in that.
Yeah, we were hoping we would see something like that, but so far what we've seen is more consistent with the idea that there's just more PAL2. We've looked at different perturbation studies from others, and we basically see that when you do the perturbations, things go up proportionally, it seems. Not quite proportionally, but proportionally enough that we think that it's just technical variation.
And then in the second part with where you have the beautiful kind of bubble assay for the polymerase, if you look at some of the other histone genes where you don't, the H3 and H4 genes where you don't have the head-to-head arrangement of the genes, do you see NPAT and the bubble sitting at both the H3 and the H4 genes?
Yeah, we do. We do. The thing is, when it's bidirectionally oriented, it's constrained. If there's not another histone gene going the other way on the other side, then the signal sort of smears out upstream. So that's why we basically like to focus on that. But we basically see the same thing. It overlaps the 5' end in the upstream region, and NPAT is right. NPAT and PAL2 are right together. They're right together everywhere we see it. And this gives me the opportunity to bring up one other thing. It turns out that we also see it outside the histone locus body, and we see it within the histone locus body outside of the histone genes. We see this correspondence of NPAT, even though there's NPAT and PAL2, even though there's no transcription there. But where we see it outside is on chromosome 6, and that means it's linked to the major histone cluster. So what we think we're looking at is in 3 dimensions, the proximity to the histone locus cluster, because there's just a lot of NPAT there.
Okay, Luai, do you want to turn your mic on?
Thanks, Dr. Henikoff. That was awesome. I had a two-part question as well. So you talked about the microRNAs coming from the acrocentric uh, parts of the chromosomes, and I was wondering if you could comment on, whether you thought that was suggesting that Pol II was in the nucleolus, or if I misunderstood that part and you were and
I'm grateful for that question because this is something we haven't worked out. So we've been going up and back as to whether it's PAL1 or PAL2. There's evidence, depending if we look at PAL2 factors. So this is all discussed if you go to the bioRxiv paper. But anyway, the what's puzzling is that these are the so-called spacer promoters. They were first described in Xenopus and have been the spacer promoters. This is exactly where we see it over the spacer promoters, and it's been a puzzle for 50 years what the spacer promoters are doing. We only see this in human. We don't see it in mouse. Now, we don't. There's some evidence that tells us that it's PAL2, but other evidence says it's not PAL2. We know it's not PAL3 for another reason, and that's also in the paper. I didn't explain that. So whether it's PAL2 or PAL3 is something that we really haven't decided on. I tend to favor PAL2, but I really can't say that. See, it could be most of the ribosomal genes are inactive, so it could be on inactive copies where we're seeing this, and not on the active copy, so there's no interference. And then CTCF is on both sides, and it makes a little loop with what we argue is parallel CTCFs, and basically makes a little loop that protects it from everything going on. So I have to, yeah. So I think it's PAL2, but then again, I could be wrong. It could be PAL1.
yeah, I I'll email you about this afterwards. But the second part of my question, that's fascinating. That's actually really cool. So as you alluded to, the microRNAs are really strongly regulatory of like total protein levels. Have you considered, or is this something in your data where maybe a lot of those microRNAs are gating the amount of histone protein? So you had like um, essentially a feedback loop of the total amount of protein being regulated by
Yeah.
RNAs regulating the histones. And so you essentially had like a transcription to translation gating.
Yeah, I suppose I hadn't thought of that. That's something we maybe should look into. Yeah, but we, but so far the evidence is that what's sufficient is H4 feedback regulation. One thing we like about that is H4 feedback. So H4 has been around as long as the histone gene's been around to the origin of eukaryotes, and we can say the same thing about PRMT5. Although it's sometimes lost in some lineages, it can be dated back to the origin of eukaryotes. The problem is we really can't make that argument for these microRNAs. We see them in primates, but we don't actually see them in mouse. So I would just sort of make a evolutionary argument that it seems unlikely, but it might be worth looking at, sure.
Okay, if you've got time for a couple more questions, there's two more in the chat.
Oh,
Um,
oh yeah. Well, there are a couple of questions here. Let's see. Okay, I'm not sure I understand the question. Why? Okay, is there a critical level of H3H4 excess beyond which the centromere integrity collapses? I can just refer to a paper from Simone Giunta and others came out in 2021, where basically they were the ones that showed that if they knock down CENP-A, they basically see these replication transcription conflicts that are causing that are causing centromer breaks. It's not an imbalance. It's basically it's indirect evidence. So I don't know about imbalance. That's our model. That I really don't have any evidence on that. So why does it lead to whole chromosome arm loss rather than focal copy number change of chromosomal rearrangements? Well, that's because it's the centromere that breaks. And the centromere can only give, if centromere breaks, will give you the whole arm changes. And that's where 80%, according to Cancer Genome Atlas, 80% of aneuploidy is that. So you might think about focal amplifications being common. They're actually quite uncommon relative to this level, this particular type of what's called chromosomal instability. So when you see the hallmark of cancer is chromosomal instability, 80% of it is this. Okay, so there are focal amplifications occurring, but it's not on our radar. Okay, next. Oh, another question. Do you ever? Okay, CENP-A. Yeah, do you ever find it in front of replication independent histones? Oh, that's an interesting question. No, we don't. We don't see it there. We don't see it in. So the replication independent histones seem to be completely independent as far as we can tell of this whole process, except that they are regulated by PAL2 and they're regulated by levels of histones. So they H3.3 has to replace histone canonical histone H3. Does this suggest that NPAT contains DNA binding motifs there? No, NPAT doesn't have DNA binding motifs. It's completely unstructured except for a few alpha helices. And those alpha helices, according to AlphaFold, actually interact with H4. So but that's not part of DNA binding. That's the interaction with histone H4. All the rest of it is unstructured. That's kind of what makes us think that NPAT is really being held by the weak interactions between the CTD and the unstructured regions. And that would allow NPAT to be released and everything to get released caused by electrostatic repulsion. So each phosphate that gets put on, gets put on a serine, threonine, tyrosine, each one at neutral pH is a negative charge of 2, almost 2. And so that's a lot of negative charges for all of these phosphates. So anyway, so that's what's driving our model. Yeah.
Hey, and one more just popped into the chat. Or a popular man.
Okay. Okay. How do you say? Oh, H4 binding the chaperone. We don't know the chaperone. We think it could be any of the various, anything that's been seen. There are histone chaperones that will chaperone single histones, particularly H4 has various chaperones. So we know that they've occurred. They've been seen in other organisms as well. But we don't know what chaperone might be doing it, or maybe a bunch of chaperones. Yeah, it's a good question. I don't have a good answer.
Okay. I see one I'm not sure we got to about
Oh,
Paul 2 Chung Gil. I don't know if you want to just maybe turn the mic on. It's right above Andrew's question.
Chunga. Oh, okay. Alternative transcription start sites drive disease progression. Have you applied your workflow to Oh, is this better than fresh tissue? So actually, the AML I referred briefly to AMLs. We don't do those. Of course, those are like cell lines, and the resolution we get is just as good. The resolution is very similar to what we get, but to what we get with the FFPEs. The signal noise, I think, is probably a little bit better for the FFPEs, but it works fine. The method works fine even on ordinary cells, and we've done quite a bit with that. Yeah. Well, that's our standard CUTAC method that we applied in a lot of cases.
Okay. Well, we're about a quarter past the hour, so there's no. Oh wait, do we have one more? Oh, it's an applause. Okay,
Thank you.
mistaking applause for hand raising. Okay, well, if that's it for questions, I will just say thank you so much once again. This was super fantastic. We'll have the recording of this and the Q&As up on our YouTube channel